I would like to really to go from, as we are used to say, from bench to bedside. This is the general outline of my talk, and we will start to describe why it is important to detecting consciousness, and we will start actually from a practical problem that many neurointensivists and medical doctors, especially neurologists, are facing every day in, for instance, ICU. So after severe brain injuries, regardless the etiology, after severe brain injury, a patient can falls into a coma state, which looks more or less like this. So the patient is lying with his, in this case, eyes closed, and it basically, as per the definition, by definition, the coma is a sort of deep sleep from ancient Greek. Then the patient can, since arousal systems are redundant, the patient can open his eyes, but still does not recover necessarily the ability to interact with the external environment. And the only movements he can express in this case is a male are reflex movements.
Is the same patient?
Yes, yeah, the same patient, and will be the same patient also for some of the measurements I'm going to show in a while. And the patient also recovers some sleep-wake cycles. Okay. Now, then patient can go from coma to vegetative state or unresponsive wakefulness syndrome to states in which subtle signs of interaction with the external environments are expressed. And these are called minimally conscious states. There are at least a couple of them. And eventually the patient can emerge from minimally conscious state. Now, how do we assess? And this is the practical problem I was referring to a few minutes ago. How do we assess today in an ICU, or better in a subacute ward, or in a rehab center, the level of consciousness, and how do we diagnose these steps you saw in this trajectory, clinical trajectory? We basically assess them through scales, behavioral scales, that investigate different dimensions of the human behavior, like responses to auditory stimulation, visual stimulation, eye closure, and so on and so forth. Now the problem is that when you have massive damages to the cortex, or the thalamocortical system better, you can have also, these systems are subserving in everyday life, sensory functions, motor functions, and executive functions too. These functions can be can undergo deficit, and this of course is making the behavioral assessment of consciousness deceptive. There is an extreme case, you maybe heard about that before. The case is the one of the locked-in syndrome, in which you have basically the truncation of the motor pathways that connect the primary motor cortex to the peripheral muscles, and the patient is fully conscious but cannot express any kind of intentional movement, maybe also after the operator is asking him to follow command, squeeze my hand or move your eyes on the right or left side. And this, of course, can be a good example on how the behavioral assessment of consciousness can be deceptive. That's why, since say 2006, more or less, many brain-based and not behavior-based markers have been devised by several groups around the world. And there are, we can go through that. We don't have time for that, but basically the operator, this is the seminal work by Adrian Owen. The operator is asking to imagine to do something like playing tennis or walking around your home, then by the fMRI scan, the patient was showing exactly the same pattern of activation that was shown by healthy subject during the same task. And there are different versions of the same paradigm now, using also EEG, spontaneous EEG. There is an alternative approach, which is based on, which stems from the importance of complexity, for brain complexity, for consciousness, and this is the very first paper that linked these two concepts, now in 1998, authored by Giulio Tononi and Gerald Edelman. What these two authors meant by complexity. Complexity is, in this case, the optimal balance between integration, the ability of different cortical areas to interact, and differentiation. These cortical areas have display some functional specialization. And actually, recently we have published this meta-analysis in which we went through the papers published after the paper by Giulio Tononi and Gerald Edelman, and we identified 182 papers after their time that are linking consciousness and complexity across, we using different techniques and across different scales and also animal models. We came up also to show that there are basically three approaches to measure complexity. One is based on topological differentiation, one is based on temporal differentiation, and the third one, the one we chose in our lab, is based on perturbing the brain and recording what happens after the perturbation. We do that by combining transcranial magnetic stimulation, that is a non-invasive technique that perturb cortical areas in humans, and electroencephalography. And then by translating into a binary matrix the cortical responses to TMS, we compressed through the Lemp-Lenziv algorithm the matrix generated by the perturbation, and calculated the PCI that was already so nicely presented by the speakers that preceded me. Now, the prediction is stemming also from some theoretical consideration that came after the paper by Tononi and Edelman. The prediction is that when the system is not integrated, is modular, you have local and simple responses, and PCI should be low. On the other hand, you can have no differentiation, so you have a fully integrated system, but with very homogeneous connections, and you basically have a simple and widespread response. And also in this case, PCI is low. In the third case, when the system is both differentiated and integrated, responses are different across different measuring channels, and this will produce high PCI. Now, let me spend a few words about TMS-EEG. Since we are in an educational frame, it's also good to highlight that, of course, TMS for people who are not familiar to that, is a technique that exploits the electromagnetic principle, induction principle by Faraday. An electric current, time varying, is passing through a coil, which is generating in turn a magnetic field, and this magnetic field is generating an electric field that excites neurons that are underlying the coil. And the other technique is an old one, is coming from the beginning of the last century and was devised by Ansberger and was in a way validated by the Nobel Prize Edgar Adrian. Now, it's not enough, unfortunately, to have a TMS in your lab and an EEG and combine them to record reliable EEG responses to TMS. There are many things it's not a plug-and-play technique, in other words, and there are many things you have to put in place in order to be confident to your TMS EEG measurements. I strongly advise people that want to approach this technique to watch this lecture by a colleague of mine, Silvia Casarotto, that is showing what are the procedures, the experimental procedures you have to apply in order to get rid of many artifacts and confounders that you can face during TMS EEG measurements. And on the other side, you can go through this other movie that has been shooted in Charleston during the last brain stimulation conference, in which our lab, together with other 2 labs from US, was running a live TMS EEG demo session, so you can see in real time how the data I'm going to show you in a few slides are acquired, and how you can have in real time an idea of what's going on in the brain of the patient you are measuring from the signals. And these are the 2 crucial steps you have to care about in order to, again, record reliable EEG responses to TMS. This is a noise masking tool to cover the noise that is produced by the coil while you are stimulating the brain, and this is a visualization tool that you have to put in place in order to have in real time to perform a quality check of the data you are going to record. This is our lab today, and this TMS coil, figure of 8 for the aficionados. This is a neuronavigation system that is able to, that allows to target a cortical spot and keep it constant during the experimental session. And this is the response of the brain. In this case, based on my experience, I can say that it's a stimulation of the premotor cortex, more or less here. These are the 60 EEG channels, and here you can see the TMS evoked potential in real time. If you apply these procedures, this is the extra challenge of the day. So you have to touch the left side of the mouse pad.
I can move the screen a little.
Okay. Got it. So here you have 4 cortical spots stimulated: the premotor cortical area, motor cortical area, parietal cortical area, occipital cortical area. You can see already from the waveforms that they are different. This is also a way to look at the differentiation, measuring differentiation of different cortical areas. It's not only something related to function of these different cortical areas, and also in terms of scalp topography, you can see how different are these responses also at late latencies. This movie I know already is not going to work, but let me spend a few words about that. There is an ongoing debate about the influence that sensory afferents can have on TMS evoked potential, but we are now with a super controlled study. We are comparing the responses to premotor stimulation to primary motor cortex stimulation with electrical scalp stimulation at different intensities and the auditory stimulation. And the bottom line message is that while these responses are so different between them, and the scalp sites where we were stimulating were the same here, here, and here, you can see how basically increasing the intensity of stimulation of the electrical sham stimulation, you get always the same response. And here you have the auditory, the response to the auditory stimulation, which is, again, not differentiated between the two areas and can be nicely controlled by the noise masking tool that I've shown you a few slides ago. Now, if we stimulate different cortical spots, you have different waveforms here: occipital cortex, parietal cortex, premotor cortex, and if you stimulate the premotor cortex in the same subject while the subject is falling asleep, you can have, come on. So here you have the reconstruction of the cortical sources that are underlying these scalp potentials. You can see how the response is moving from the spot that was directly stimulated to other areas on the contralateral side or on the same side but on different lobes. Here the Marcello was stimulating when he was working in Giulio's lab the premotor cortex, and the response was moving posteriorly. You can capture that in just one picture in which you can see the time color-coded and how many areas were engaged by the stimulation. Now, what happens 15 minutes after when the same spot is stimulated and the subjects fell into deep sleep? You can see that the response is local, stays there, it stacks there. It's not going around, and the significant part of this response is lasting half of the original response during wakefulness. We have applied this technique during deep sedation and anesthesia with different anesthetics, and we came up with this sort of scenario in which when the subject is conscious, in a way these were sort of confirmations to the predictions we have done. So the responses are complex over in time and space when the subject is conscious, while they became local or widespread, but always simple and stereotyped when the patient is anesthetized. I will skip because I have a few minutes left. And then we measured PCI in all these conditions. We calibrated actually these two bunch of these two pools of data were our calibration sets, and we then moved to patients. This was the first study in disorders of consciousness patient that was confirmed a few months after by another Italian group. Here you can see how in vegetative state patients the responses are local and simple, while in minimally conscious state patients and locked-in syndrome patients, the responses are complex. And this is nicely revealed by PCI that shows a high sensitivity to detect MCS patients. Also, when the spontaneous EEG, and this is something interesting also for Mavi and Alain, when the spontaneous EEG is not showing a slow activity. So PCI is low, and you trigger a slow wave even when the EEG is not prone to do that. We ask then, by focusing on these patients with a sleep-like response, that was not going away when the subject was awake. We ask what can be the mechanisms behind that. This was already mentioned by Mavi. We think that behind the collapse of complexity and so hopefully of consciousness, you have these off periods that actually showed up also in our TMS EEG recordings. This is a paper by Andrea Pigorini that will talk tomorrow more extensively about these and other studies that we detected both in vegetative state patients and healthy subjects that were deeply sleeping. And this is also a feature that was not that was kind of preserved despite the spontaneous activity that was ongoing in that moment. These are the studies already shown by Mavi. Let me add also this study by the Johan Storms group, in which in animal models they were basically confirming the observation we have done in humans through TMS EEG. There are plenty of questions opened by animal studies. These are studies done in anesthesia in monkeys and mice, and it's a way to say we still need animal research to deeply understand the mechanisms behind the loss, for instance, of complexity and consciousness in humans. And also, going back for a while to TMS, we need them also to understand what are the real targets of TMS in a living brain. And again, to understand mechanisms, we need an exploration across scales. And again, Andrea Pigorini will talk about that tomorrow in his talk. I'm just a few slides. I'd like really to show what happens in patients that emerge from MCS. Here you can see how the local response, these are the responses of cortical sources to perturbation re-emerge when the patient was recovering consciousness, and how PCI is nicely following this trajectory. But I will also that the off periods or cortical bistability is disappearing, and is leaving the room for a sustained electrical response to TMS when the patient is emerging from the MCS. But we also have observed many patients that were behaviorally VS, so in a vegetative state, with high complexity responses. This is just an example. This is a patient with a very abnormal spontaneous EEG. This is the coma recovery scale. The patient was in the vegetative state or unresponsive wakefulness syndrome state, and yet PCI was showing here in these very early days high complexity values. The patient was fluctuating, but PCI was kind of stable even when the EEG was showing some epileptiform activity. And by the way, in this patient, one of the paradigm I've shown at the beginning of the talk was applied, and the patient was showing brain responses to command. We have now a few of these examples, and let me focus on one of these. This is a patient that was showing high levels of PCI when we were stimulating the parietal cortex, and while the frontal cortices were still in a low complexity state. Then the patient was still in a vegetative state, but both areas were recovering complexity. And finally, the patient, after 3 months, was recovering consciousness. But in a way, PCI was able to detecting the capacity for consciousness that was unexpressed in those days and was finally expressed. This is the patient in this day, and is clearly able to follow commands. So he's asking to move the first finger. The middle finger, the thumb now.
Sempre più difficile, bravissima, l'ho visto il movimento, ottimo Jessica. Riesci a muovere l'anulare? Grande. Ottimo, bene Jessica, l'ho visto benissimo il movimento. Fermati, se riesci adesso il mignolo. Spettacolo. Grande, bravo.
Okay, let me thank the patients or their families, and this is really a choral work. Of course, we are under the guide of Marcello Massimini, but there are plenty of co-workers around the world now that allow to reach these results. Thanks a lot.